Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Welcome, everybody. Some procedural stuff before we get started. So this is going to be our last webinar of the calendar year. Next webinar will be January 14th, and that's going to be Lorenz Lahm from Helmholtz Munich, and he's going to be talking about membrane V2. If you have questions during the webinar today, please feel free to either send them to one of the hosts through chat or to use the Q&A feature. And with that, I'd like to introduce Ariana Peck from Chan Zuckerberg Imaging Institute, who's going to be talking about Ariotomo 3, or I guess I should be calling it Ariotomo Live. So with that, Ariana, thank you for joining us, and all yours.
Great, thank you. Thanks for this opportunity to describe some of the tools that we've been developing at the Chan Zuckerberg Imaging Institute. The focus of my talk today will be Artomo Live, which is a collection of 2 software packages that we've developed to perform the first several steps of data processing for cryo-electron tomography data in real time and at high throughput. Artomo Live is a key part of our broader effort at CZII to transform cryo-electron tomography, or cryo-ET, from a relatively niche and specialized technique to a routine method to probe cell biology. Our motivation for doing so is that so much of cell biology is still uncharted territory, and cryo-ET uniquely enables us to peer inside cells and visualize molecular interactions with up to sub-nanometer resolution. In this video I'm showing here, one of my colleagues is navigating a cell that's been imaged by cryo-ET and labeling various cellular components and proteins of interest to deepen our understanding of its molecular architecture. If we can identify sufficient copies of a particular molecule of interest across multiple tomograms or 3-dimensional maps of the cellular sample we imaged, then we can average these copies together in a process called subtomogram averaging, or STA. And this averaging procedure enables us to overcome the inherently low signal-to-noise ratio and missing wedge of information associated with each individual molecule to determine a consensus structure to higher and in some cases sub-nanometer resolution, importantly while still maintaining information about these molecule cellular context. But achieving sub-nanometer resolution can be quite challenging, especially in the crowded environment of the cell. If we take a look at all of the in situ subtomogram averages that have been deposited in the electron microscopy databank, we can see that it's really only been in the past few years that we've been able to accomplish this at all. Overwhelmingly, these sub-10 angstrom structures are of ribosomes, which are highly abundant, and of viral and filamentous assemblies, which tend to benefit from high symmetry. A key reason why extending this workflow to new molecular species is so very difficult is simply the challenge of scale. As you can see in this plot on the right here, we often need to locate tens of thousands of copies of the molecule of interest, usually across tens to hundreds of tomograms, for high-resolution STA to succeed. As a result, throughput has been a key consideration in developing Artomo Live. Thanks to parallel data acquisition schemes like PaceTomo, we've been able to routinely collect over 1,000 tilt series per week on a single Krios. But realizing the full potential of this accelerated data collection requires that data processing be able to keep pace. Ideally, we'd like to provide users with tomograms in real time so that they can adjust data collection as needed and immediately start the downstream tasks of particle picking and STA. A second key consideration has been reconstruction accuracy. Biological samples tend to warp and deform upon exposure to an electron beam, and the signal from our sample will be further delocalized by the contrast transfer function, or CTF. Left uncorrected, these motions and aberrations severely limit the high-resolution signal that's contained in our data. A final consideration has been contrast, because the extremely low contrast of cryo-ET data can be prohibitive to downstream analysis. But enhancing contrast comes at the expense of high-resolution signal, so any denoising we perform must carefully balance these competing needs. With these considerations in mind, we developed Artomo Live to automate tomographic alignment, reconstruction, and contrast enhancement for use during live data collection. This slide shows a schematic of the Artomo Live workflow, which is composed of 2 GPU-accelerated packages. The first of these is Artomo 3, which carries out all of the tasks indicated by the dark blue arrows as each raw tilt movie comes off the microscope, and that will be the focus of the first part of my talk. Afterwards, I'll describe the second package, DeNoise-It, which runs in parallel with Artomo 3 to train and then apply a denoising model to the tomograms that Artomo 3 continually writes to disk. Focusing first on Artomo 3, the software provides a streamlined workflow to reconstruct tomograms from tilt movies and can be divided into 3 modules. The first module corrects for beam-induced motion in 2D to generate the motion-corrected tilt stacks. The second module then performs tomographic alignment and CTF estimation. While it's simplified in the schematic here, this is actually an iterative process in which the per-tilt CTF parameters are first coarsely estimated prior to computing the global alignments that define the tilt axis angle and correct for the relative translations between the tilt images. The CTF parameters are then refined before computing the local alignments that correct for the 3D deformations that occur in subregions across the full tomographic volume. The third and final module then applies these global and local alignments to the tilt series in order to reconstruct tomograms, either by SART or weighted back projection with a local CTF correction optionally applied. In addition to tomograms, Artomo 3 saves various other output files to facilitate downstream processing. These include a record of the global and local alignments applied during reconstruction, the unaligned tilt series and CTF parameters that are needed by many SDA packages, and then finally a collection of quality metrics that we found quite useful for data curation. For those familiar with Motion Core 2 and Artomo, Artomo 3 unites these 2 software packages into a single integrated pipeline. One reason for doing so was efficiency, since integration reduces the number of times we have to read large data files from disk. A second reason was consistency, because the use of a single convention across the full pipeline avoids issues that arise from different software packages frequently adhering to different coordinate conventions. This can be particularly problematic for tomography data, where a change in coordinate convention can very easily flip the defocus and/or physical handedness of the data. Finally, we wanted to reduce barriers to entry and save users from having to write custom scripts simply to transfer data from one software package to the next. But Artomo 3 is much more than just the seamless integration of these 2 software packages, and over the next several slides, I'll highlight several new features that we've introduced to improve throughput and accuracy. One of these new features is multiple entry points, which are indicated by the red arrows here. While it's useful to have a pipeline that can run from start to finish without interruption, we recognize that in many cases, users will want to reprocess data from an intermediate state, for instance, after adjusting an alignment parameter like the number of local patches, or a reconstruction parameter like the volume thickness. These multiple entry points allow users to bypass the upstream processing tasks and more quickly generate a new set of tomograms based on the adjusted parameters. A second new feature automates the measurement of sample thickness. To estimate thickness, we first reconstruct heavily binned tomograms and then compute their cross-correlation between adjacent slices along the tomogram's depth. An example correlation profile is plotted in yellow here, where it's overlaid on an XZ slice through this tomogram. These correlation profiles tend to have characteristic shapes, with higher values in the central region where the sample is most concentrated, that then decay to minima beyond the sample boundaries. The correlation scores shoot up again towards the tomogram edges due to back projection artifacts, but those we can safely ignore. Focusing just on the central region, we determine the sample boundaries based on where the correlation falls to the value halfway between the peak nearest the center and the minima on either side. It's a bit difficult to see in this XZ projection here, but if we take a look at this 3D view of the tomogram below, we can see that these detected boundaries do a very good job of fully encompassing the sample while excluding the vacuum on either side. We found that this approach generalizes well to diverse sample types, but one caveat is that it assumes that the sample is leveled within the tomogram. In this middle panel here, for instance, we see that while the boundaries are correctly detected, there will still be a lot of vacuum in the reconstruction due to the large alpha tilt, which is a characteristic feature of lamella samples like the one shown here. Artomo 3 automatically accounts for the alpha tilt offset during the alignment process and can optionally correct for it during the reconstruction as well, as shown in this panel on the right. A second caveat is that ice contamination can confound the detection process. In this panel on the right, for instance, we can see that while the upper boundary is correctly detected, the lower boundary is not due to the presence of this large ice crystal that's indicated in red. Correctly measuring the sample thickness is important to improve alignment accuracy. In the case we're looking at here, the sample thickness is roughly 1500 angstrom. And when we use that value during the alignment process, it yields the best reconstruction with the highest spatial coherence, which is shown in this middle panel here. But this value falls in a bit of a Goldilocks zone. And if we use a value that is too thin or too thick during the alignment process instead, the reconstructions have deteriorated quality, as shown in the left and the right panels. And this is particularly evident in the regions that are outlined in white, where we can see that we lose our ability to resolve some of these synaptic vesicles and the membrane features in these tomograms. A third new feature estimates and locally corrects for the CTF. CTF estimation is performed by dividing each tilt image into overlapping patches and then computing the power spectrum of each patch. These power spectra are then rescaled to compensate for differences in defocus across the tilt image, following an approach that was first implemented in BSoft. This spectral rescaling increases the spatial coherence of the average power spectrum, which we then use to determine the CTF parameters. Though at high tilt angles, we only refine the defocus values and bootstrap other parameters like the astigmatism to the nearest tilt image to improve the optimization. As you can see on the right here, this approach has enabled us to robustly fit the CTF across a range of defocus values and also at high tilt angles where the signal-to-noise ratio is very low. We then use these optimized CTF parameters along with our refined measurements of the sample geometry in order to locally correct for the CTF in a way that accounts for the defocus gradient, which is a topic that I'll revisit later in this talk. A final new feature that I wanted to highlight is a collection of quality metrics that Artoma 3 now provides for every tomogram it reconstructs. Our hope for these quality metrics was that they could assist or even replace the very time-consuming visual inspection that we often perform when deciding whether to keep tomograms for downstream analysis. From visual inspection, it's often immediately obvious whether a tomogram is of high quality and well-aligned, as in this example on the left, or is poorly aligned, as in the example on the right. But visual inspection simply doesn't scale to when our data collection sessions span hundreds or even over 1,000 tomograms. To address this bottleneck, Artoma 3 now tracks various quality metrics that are shown in these plots here. And in the future, we plan to extend this to track additional quality metrics as well. 3 of our current metrics, specifically the tilt axis, the global shift, and the fraction of bad local patches, report on various aspects of the alignment process, while 2 others report on the quality of the CTF fits. The remaining 3 metrics from our current set describe various characteristics of the sample geometry, including the sample thickness and the alpha and beta tilt offsets. And as I'll describe later in this talk, we found these quality metrics very useful for streamlining and automating data curation. We've also found these metrics informative for comparisons across different data collection sessions. In these plots here, we're tracking these quality metrics across 11 different data collection sessions where we were acquiring data from samples of purified synaptosomes. And tomograms from 2 of these data collection sessions are now available on the CryoET data portal for anyone interested. When we inspected these plots, we noticed that session 6 in particular seemed to be a bit of an outlier in terms of its quality score. And when we looked back through our notes, we realized that during this session, we had targeted sites that were too far away from the tilt axis, which is an issue that we addressed in later sessions. And so in addition to aiding data curation, we envisioned that these quality metrics could provide a useful diagnostic for how choices we make during sample preparation or data collection impact data quality. Switching gears, the final portion of my talk will focus on the second component of Artoma Live, which is a denoising routine that provides high contrast tomograms. We were motivated to integrate denoising into our live processing pipeline because in almost all cases, some type of contrast enhancement is needed, not just for visual interpretation, but to aid particle picking and segmentation as well. It's easy to see why this is the case if we inspect tomograms that have been reconstructed by weighted back projection without any additional filtering applied to them. In this example here, we're looking at a 5-angstrom thick slice through a tomogram of purified proteins and cellular lysate. And even though the sample was well behaved and the tilt series aligned quite nicely, it's still really difficult to discern what's happening in this tomogram simply because the signal-to-noise ratio is so incredibly low. Here's the same slice through this tomogram after we've denoised it. And as you can see, there's really a night and day difference in terms of what we can infer about the contents of the sample and the distribution of particles within it, thanks to this denoising. To perform denoising, we've been leveraging a machine learning algorithm called noise-to-noise. This algorithm is a bit older by the standards of the machine learning field, but it's really well suited to our problem at hand. Noise-to-noise relies on a convolutional neural network with a UNet architecture, as shown in this schematic. But really the salient point here is that this neural network provides us with a function that has sufficient representative capacity that we can train it to take in our very noisy and complex structured tomograms and have it generate cleaned up versions of those tomograms in a way that better preserves structural features than a simpler denoising approach like a low-pass filter could achieve. Training our neural network to learn this mapping between noisy and clean tomograms would be straightforward if we had access to pairs of noisy and clean measurements, because in that case, we could apply our neural network to the noisy measurements and then compute the loss or residual to their corresponding clean counterparts. And the gradient of this loss function would point us in the direction that we should adjust the 3 million free parameters of our neural network in order to optimize this mapping between noisy and clean. But in the case of experimental CryoET data, we never have access to the clean measurements, and the cellular samples we're imaging and the aberrations and non-idealities introduced by the imaging process itself are sufficiently complex that it's really challenging to generate realistic simulated data. The key insight of the noise-to-noise algorithm is that it suffices to have access to pairs of noisy measurements in order to learn this mapping between noisy and clean. And in the case of cryo-ET data, we are in this situation because every image we collect is composed of a series of frames, all of which are imaging the same underlying field of view, but they differ because the noise is stochastically generated. So intuitively, it seems like we should be able to infer what the signal of interest from our cellular sample is, because that contribution should be consistent across all of these frames and only the noise should differ between them. The noise-to-noise algorithm provides us with a framework to do exactly that, because even if you consider any individual pair of noisy measurements, the gradient of the loss function is not guaranteed to point in the right direction, but the average gradient will. As a result, we should be able to learn how to map these noisy measurements that we are able to collect to this unobserved clean target that we're never able to measure. Noise-to-noise has previously been implemented for cryo-ET data in software packages such as CryoCare and Topaz Denoise, but we were motivated to re-implement this algorithm, first to streamline its integration with Artoma 3, including during live data collection, and second to explore ways to improve its effectiveness, and in particular to retain more intermediate resolution features that denoising tends to suppress. For integration purposes, we've tasked Artoma 3 with generating the paired noisy measurements, in our case paired tomograms, that are required to train the neural network. Artoma 3 accomplishes this by splitting the motion-corrected movies into half sets, one of which consists of all of the even frames, while the other contains all of the odd frames. These half sets are then summed separately in order to generate the corresponding even and odd tilt stacks. Because the full tilt stack contains all of the frames, it has the most signal, so we use that to determine the tilt series alignment parameters. The set of refined parameters is then applied not just to the full tilt stack, but to the even and odd stacks as well, in order to generate the corresponding tilt series, which are then reconstructed into the corresponding tomograms. The even and odd tomogram pairs are then used to train the neural network, and once fully trained, this neural network is applied to the full tomograms in order to denoise them. It's worth noting that there are other ways that we could have gone about generating the paired noisy measurements that are required to train our neural network. For instance, instead of splitting our data at the frame level, we could have performed the splitting at the tilt series level and separated our tilt images based on whether they were collected at even or odd tilt angles. The disadvantage of this approach is that it results in sparser sampling across the collected tilt range, which would then be propagated into the even and odd tomograms during reconstruction. And the artifacts introduced by the more severe missing wedge of information between the tilt angles would likely be picked up on and reinforced by the neural network. Another approach we could have taken would have been to split our motion-corrected frames either randomly or based on whether they are acquired during the first or second half of the movie. But ultimately, it's adjacent frames that share the highest degree of structural similarity and accumulated dose to one another. So really, it's by following the framework that I described on the previous slide that we're best able to satisfy the premise of the noise-to-noise algorithm, because this even-odd splitting at the frame level generates half sets with the highest degree of structural coherence, which we then preserve by applying the same alignment parameters during reconstruction. I wanted to provide a detailed explanation of how we generate our training data because empirically we found that the way we pre-process our data really matters and can significantly impact the quality of denoising we're able to achieve. One way we've observed this is through data curation. From the start, we've selected training data based on the assumption that training with the highest quality tomograms would yield the best results. Initially, we performed this curation based on visual inspection, but now we leverage the quality metrics from Artoma 3 that I described earlier. This has made the curation process not only more objective, but also feasible for large data sets, like this data set of a few hundred tomograms of purified lysosomes that we're looking at here. In this case, only tomograms that met certain thresholds for all of these quality metrics were included in the training process, and those 34 high-quality tomograms are highlighted in blue in these plots on the left. And when we trained on these data, we found that the resulting denoising was not just effective at increasing the contrast of these membrane proteins that line the perimeter of the lysosome, but also quite consistent as well across a range of values for this model hyperparameter n, which is the number of subvolumes extracted per tomogram during each training epoch. And this consistency is valuable because it means that we don't have to perform an extensive hyperparameter search in order to achieve good denoising results. By contrast, training on low-quality data results in poor denoising. The 34 low-quality training tomograms highlighted in red in the plots on the left were selected based on having low CTF scores or poor alignments. And when we trained on those data, we found that for smaller values of n, we tended to observe under-denoising, which insufficiently flattened the background. And for larger values of n, we consistently observed over-denoising that washed out some of the smaller membrane proteins around the lysosome and also introduced patchy artifacts into the background as well. While these results weren't surprising to us, it was still useful to verify that we could leverage our TOMA 3's quality metrics in order to select training data that would yield robust denoising. But this also raised the question of how much low-quality training data denoising can tolerate. To address that question, we next trained on the first 34 tomograms that were required during this data collection session to mimic the scenario of running denoising live, except without any data curation. What we found is that the model effectiveness is actually less sensitive to the quality of the training tomograms than we had anticipated. In this lower panel here, for instance, we don't see the very severe under or over denoising that we observed on the previous slide. But the results are still less consistent than training on high quality tomograms. And we do see some less effective denoising, like in this lower center panel here, for instance, where some of the smaller membrane features are actually blurred into the background. So it seems like some degree of data curation would actually be useful to ensure to maximize consistency across hyperparameter space, but that we don't need to be super stringent during the selection process. Given the stochastic nature of model training and the high variability of cryo-UT data, it may not be possible to come up with hard and fast rules about specific thresholds that we should set for these quality metrics. But this is an area that we plan to explore further to better automate data curation in our live processing pipeline. As with data curation, we found that the way we correct for the CTF can impact the quality of downstream denoising. In our initial implementation, we followed the strategy taken by ISONET and WARP, which deconvolves the CTF by applying a Wiener-like filter to the tomograms using a single defocus value. But this approach ignores the defocus gradient, which in turn will reduce the spatial coherence of the CTF-corrected tomograms that we use for both training and inference. This motivated us to implement a new CTF deconvolution in our TOMA 3 because we were curious to see if a more rigorous CTF correction would improve downstream denoising. In contrast to ISONET, our TOMA 3 deconvolves the CTF from the tilt series rather than the tomograms, so that it can account for the tilted geometry that gives rise to the defocus gradient. One of the data sets that we used to compare these two strategies was the lamella of human cilia, which is shown on this slide here. As you can see, without any denoising, it's pretty much impossible to discern these proteins that are embedded in the membrane and arrayed along these filaments. When we compared these two preprocessing strategies, we found that the more rigorous CTF correction implemented in our TOMA 3 enabled subsequent denoising to retain more intermediate resolution features. And that's particularly evident in these inset regions here, where the proteins in the membrane and along these filaments are better resolved in the R-TOMA 3 case. We wanted to provide so I guess we wanted to move beyond mere qualitative assessments and really quantify the difference in contrast enhancement that we were getting with these two approaches. Initially, we estimated contrast enhancement based on comparing the denoised tomograms to the weighted back projection tomograms in terms of their power spectra. And that gave us the estimates for contrast enhancement that are listed here in decibels. But when we dug a bit deeper, one thing we realized is that in the ISONET case, the vast majority of the contrast enhancement is actually coming from the low pass filter that's applied as part of the CTF deconvolution. And denoising provides very little contrast enhancement on top of that. And this is problematic because if we apply a very strong low-pass filter during data preprocessing, we'll end up blurring out many of the structural features that ideally our neural network would learn during training and preserve during inference. I should note that the strength of the low-pass filter applied as part of the CTF deconvolution is actually an adjustable parameter. And in the ISONET case, we did perform a hyperparameter search and found that we needed the strong low-pass filter in order for the denoising to be effective. We realized that our analysis of contrast enhancement would be more discriminating if we focused on local rather than global contrast. As an example, here's a cross-section through one of the microtubules from the tomogram on the previous slide. And what we see is that when we denoise on top of R-TOMA 3's more rigorous CTF deconvolution, in real space, we're better able to resolve the individual protofilaments of this microtubule. And correspondingly, in Fourier space, we see features out to higher resolution as well. And if we inspect the radial intensity profiles of these cross-sections, we can see that the features at intermediate resolution that we're seeing in the R-TOMA 3 case align with features that we see in the radial intensity profile of the weighted back projection case as well. And so it's not that our neural network is hallucinating these features, rather these features are there. And by applying a CTF deconvolution that is more rigorously corrects for the CTF and relies less heavily on a low pass filter, we're better able to preserve these features during denoising. For a more robust analysis of local contrast, we leveraged another data set in which we had already thoroughly identified many regions of interest, including an extensive set of virus-like particles, an example of which is shown in this upper panel on the left here. This enabled us to easily identify an equivalent number of background regions based on the local intensity statistics of these tomograms, and an example is shown in this lower panel on the left. Taking the ratio of the power spectra of the regions of interest to the background regions provides the most insight into how much contrast enhancement we're seeing in the parts of the tomogram that we most deeply care about and will actually use for downstream processing. Based on this analysis, we estimated that we were observing roughly a 5-fold increase in the contrast enhancement in these specific target regions for our denoising compared to weighted back projection. And importantly, this contrast enhancement extended out to intermediate resolutions without introducing artifacts at higher resolution as we observed in the ISONET case. As I've just described, part of our motivation for integrating denoising into our TomoLive has been to leverage the quality metrics and CTF deconvolution from our TOMO3 in order to improve contrast enhancement. Looking ahead, we plan to explore closing this loop by leveraging the denoised tomograms to improve tomographic alignment in an approach we've been thinking of as subvolume polishing. Specifically, we plan to forward project the denoised tomograms into tilt series and then use these contrast-enhanced tilt series to recompute the local alignments that correct for 3D deformations that occur in subregions across the full tomographic volume. Any improvements we get during the alignment procedure would result in more spatially coherent tomograms during reconstruction, and that would in turn provide the basis for improved denoising. So this could be performed in an iterative fashion. That's the next big ticket item on our radar, but something we found immensely valuable has been to receive feedback from users, especially about challenging data sets where the data processing could be improved. And so we'd be thrilled to hear from anyone in the community using the software about new features that we could integrate that would help drive biology projects forward. With that, I'd like to note that all of the work that I've described today has really been a team-wide effort across all of CZII, though I wanted to highlight the contributions of a few individuals in particular. First and foremost is Sean, who developed our TOMO3 and has contributed heavily to the development of Denoise It as well. Sean and David have long been committed to delivering software to our community that really helps drive our field forward. Yue and Reza have provided invaluable insights into what new features we can integrate that would help the software, enable the software to provide better data processing for a diverse range of data sets. And we've also had great discussions with Dari about contrast enhancement and with Utz about different coordinate conventions that different software packages in our field use. All of the data that I showed today was collected by Liz, Daniel, and Reza's teams and an external collaborator, Bram Koster. And then finally, I wanted to thank Bridget, Clint, and David for all of their support and especially providing not just really great technical insights, but also invaluable perspective on how to ensure that the tools that we develop are useful for the broadest community. And with that, I'd like to thank everyone for their time, and I'd be happy to take any questions.
Thank you. Thank you very much, Ariana. That was a great talk. And we have a couple of clapping emojis going. So it's not quite the same as the audience, but the best we can do over Zoom. So if folks have questions, again, feel free to send them in chat or use the Q&A feature. As usual, I have many questions. With the caveat, they may not all be good questions, or they may not even be well-posed questions, but I do have a couple. So I guess for denoise ET, am I understand, is that something where you do that for training the model for each collection, or is that you train one model and then you get a good model, you use that multiple times?
So what we found is that for data collection sessions that are collecting on very similar samples to samples that we've acquired previously, the model does work well if we've already trained it on something similar. But once we change either the sample or the data collection parameters like the defocus, et cetera, then it does make sense to retrain a new model. There is some generalizability, but you do get slightly better, or we've observed slightly better effectiveness when we retrain once the data collection or sample changes.
And from that cartoon, for a AI naive person, is that like it looks pretty similar to an autoencoder architecture. Is that like in the right ballpark intuitively?
Yeah, there are some differences. I think one thing in particular is that units rely on something that's called a skip connection, and so they don't really have a true latent space. And so it can't generate new things because it actually depends on some of the information that's being transferred from kind of the left side of the encoder or like the downstream tasks to like the kind of upstream paths. Um,
Like I said, not all of my questions were going to be well posed. Thank you.
Thanks for those questions.
Yep. And so we have a question from the audience, which could you elaborate a bit more on why you were splitting the tilt series into even and odd for introducing artifacts? Sorry,
Um,
I think I may have garbled that question. Could you elaborate why splitting tilt series stacks into even and odd stacks introduces artifacts? Which,
Right. This was actually something that was addressed when people first started applying noise to noise to cryo-ET data in the CryoCare package. And one thing so if you end up splitting the tilt images into even and odd tilt angles, then you'll have, instead of, for instance, having a 3-degree increment between the tilt angles, you'll have a 6-degree increment. And so, we already have a large missing wedge of information kind of in the space that we don't collect, but there's also missing information between the tilt images that we do collect. And so having that sparser sampling will actually introduce artifacts during the reconstruction. And it only affects the even and odd tomograms that are used for training because the full tomogram won't have that artifact.
thank you. And another one from the audience, have you considered merging high-res information with the denoise data?
Yes, that is something we've considered. It's something in particular that we actually implemented for 2D data, and we found that the results seem to work well. But it's not something that we've implemented yet for 3D data, but it is an idea that we're excited to pursue.
Cool. I guess two others. Have you considered for the denoising again, have you considered providing information from the before the CTF correction and possibly incorporating the CTF correction parameters, hyperparameters in that, or would that just like blow up the compute requirements and make it unfeasible?
Yeah, I guess we haven't. One thing that we have noticed is that, so I guess I should say that we've noticed that some degree of low pass filtering is essential during the CTF deconvolution for the noise to noise algorithm to actually work for cryo-ET data because we think it's just too noisy otherwise. And so the neural network doesn't end up kind of learning the features that we would most like it to learn. And so the strength of the CTF deconvolution is a hyperparameter that we're thinking about. I don't know that we would do it as part of I don't know during like how exactly we would implement that in order to make sure that we get kind of denoising on a reasonable timescale. But one thing we think is that there are probably certain characteristics of the sample that would indicate to us we might need to strengthen the value of the hyperparameter or the strengthen the amount of low pass filtering we apply during CTF deconvolution in order to achieve better denoising results. Though I should say that for the CTF deconvolution that's implemented in Artoma 3, we found that the same value for the strength of the CTF deconvolution actually works well across a diverse range of data sets. And it's really kind of only outlier data sets where we're thinking of adjusting it.
Well, thank you. And just FYI, that discussion kind of reminded me there's an effort by a crystallographic group who's looking into using basically deep learning approaches to learn about correcting for scaling and basically do better scaling of X-ray diffraction data sets than the traditional algorithms have. So this may have some conceptual similarities, even if I don't know if there's anything technically you could borrow.
Thanks for that point.
Yep. And all right, so we have another question from the audience. Do you have recommendations for general Ariotomo based or do you have general Ariotomo-based recommendations for extraction box sizes in terms of like ratios of your particle of interest?
Um, I guess, is that for the local alignments, or I'm not?
For subtomogram averaging. So that's an excellent question. And Ahad, if you'd like to clarify that, please do, because I'm not sure how to clarify that one.
Okay, thanks.
One. All right, so we do have, jumping back slightly, could you compare how, or could you say anything about how denoise ET compares to DDW? And would you describe it as denoising and missing wedge or denoising only?
So it's denoising without correcting for or trying to fill in the missing wedge information. So I'm not quite sure what the DDW abbreviation corresponds to, but I assume that's for a missing wedge correction, which we don't
I'm not
imagine.
sure either, and I asked about that as well. And if that clarification comes in, I will pass it on.
Oh, I think it's deep to wedge now that I think about it. Sorry.
Ah,
Yes, so we don't, we're using, we're not using that as strategy because we're not, at this point, we're not trying to correct for the missing wedge or fill it in.
right. Thank you. And let's see. Could you explain a bit more the differences between warp CTF correction tomogram reconstructions and Ariotomo 3?
In terms of the CTF correction, so I guess the main thing, I guess that one of the key differences is that warp applies the CTF deconvolution to the reconstructed tomograms. And it does so in a way that only uses a single defocus value that is applied to the entire tomogram. So it doesn't account for the defocus gradient in a way that another software package like Nova CTF does, where it's trying to account for the 3D, the defocus gradient across the tomogram. But we chose in Artomo 3 to apply the defocus or to apply the CTF deconvolution to the tilt series instead of the tomograms. Because in Artomo 3, we already have the refined geometry measurements, including the alpha and beta offsets, and also kind of local alignments and things like that, that enable us to kind of more rigorously correct for the CTF in a way that really does account for the defocus gradient. And so those, I guess, CTF corrected tilt series are then what's used to reconstruct the tomograms in the Artomo 3 case.
Thank you. And we do have some clarification on the box size question. So for how the CTF signal delocalizes in terms of box size for the particles during subtomogram averaging.
Yeah.
And so like for one example, like, 2 times your particle size, 3 times your particle size, is there a rule of thumb or any guidance there?
As someone who hasn't done subtomogram averaging, I'm not sure that I'm the best person to address this question. And unfortunately, I think I'd have to kind of think about that more to be able to answer that adequately.
I don't know is always a valid scientific answer. And, it's how science works. There's always more stuff we don't know.
Right.
Yeah, I guess one more question, which again, I apologize. This may be a naive question. It may be because I've been reading up on cryo-EM data processing. But with these denoising algorithms, is it worth doing a negative control about what happens if you try to denoise on random noise and see what happens? Or is that something that you do routinely and it's a negative control?
Yeah, we were concerned about that. And so we haven't done this in the 3D case, but in the 2D case, we did apply a model that we had trained on some proteasome data to just kind of random noise. And to our relief, we did not see proteasomes come up in the kind of denoised random noise. So yeah, I mean, I think it's a really good thing that we should more routinely kind of verify to ourselves that we're not hallucinating features with these neural network approaches.
I think everybody in grad school is probably taught about, you need to do your positive controls and your negative controls, and like you still need to do them on the computer. So
It's true.
All right. With that, I guess one more call for questions or one more call for questions or questions that I have missed. And all right. And looks like I have not missed any and not mangled too any. So thank you very much once again for presenting. That was a very great talk, and look forward to seeing more exciting tomography software coming out. So thank you.
Thanks so much.